Bioscience webinar series. This is a monthly program sponsored by Cohen Veterans Bioscience for brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through translational research. We are focused on understanding the molecular underpinnings of diseases, such as PTSD and TBI, to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk through some logistics. This program is being recorded and will be archived on the Cohen Veterans Bioscience YouTube channel and website. The presentation will be about 40 minutes in length to allow for Q&A afterwards. Please use the Q&A button to post a question. Today our speaker is Dr. Fernando E. Boada. Dr. Boada obtained his PhD in physics from Case Western Reserve University, as well as specialty training in magnetic resonance imaging at both Case Western and Harvard University Medical School. He moved to the University of Pittsburgh in 1993, earning tenure as professor of radiology. In 2012, Dr. Boada began appointments at both New York University as a professor of radiology, psychiatry, and neurosurgery, as well as director of the Center for Advanced Imaging Innovation and Research, and co-director of the Center for Biomedical Imaging. In addition to serving on numerous scientific review committees and editorial boards, Dr. Boada is a current chair of the imaging program for the International Society for Magnetic Resonance in Medicine. In this webinar, Dr. Boada will discuss the conceptual paradigm for utilizing diffusion spectrum imaging in the study of PTSD and will present current findings in the PTSD cohort. He will also discuss considerations for using DSI as a diagnostic tool. It is my honor to introduce Dr. Boada.
Good afternoon, and thank you for the opportunity to describe the work that we're carrying out with some of our group here at NYU. So the plan for today is to give you an overview and get you familiar with some of the results that we have been obtaining using the Cohen Veterans Center dataset and trying to evaluate the use of diffusion spectrum imaging as a tool for finding anomalies in structural connectivity in TBI as well as PTSD. And so in order to get us to what we wanted to do, we decided to break down the presentation as you see on the screen there. We'll give you a brief introduction about what we mean by structural connectivity, then talk about a little bit of the conceptual paradigm that we're trying to use for looking at this, some of the inherent assumptions that we make when we do this, a little description of what I call nodology. Then we will describe to you a technique that we call radial diffusion spectrum imaging that we developed here at NYU for overcoming some of the problems with structural connectivity. And then we're going to explain to you what we have dealt with the intrinsic higher dimensionality that this type of data sets entail. And we show you some results using low-rank plus sparse decomposition for looking at this data. Roughly speaking, we know that in the context of TBI and PTSD, there have been documented changes in axonal strength and integrity, and this has been shown using both DSI and DTI. And some of the areas that show up commonly are the precuneus, the forceps major, and the cortical spinal tract, depending on what kind of injury or how the injury was presented. There are connections between performance and structural connectivity that have been studied previously using neural net models. The connections appear to be suggestive, although there isn't a strong causal link right now, and we'll see as to why could be some of the potential reasons for this. And so the question that we want to pose in the context of the work that we're doing is there a causal relationship between these things? And how is this causal relationship related to cognitive function? And can we actually make a biomarker out of this? So the overall paradigm is that if we think of the brain as a computer, then we want to think about this structural connectivity as the wiring diagram for the brain. And so the implication, we hope, is that if the wiring paradigm or the wiring diagram is abnormal, then that will lead to an abnormal brain function. This particular concept is certainly not new. People have been looking at this for a while, close to 10 years right now. And some of the early work focused on using resting state functional connectivity and trying to find how the connection maps that were obtained in this setting were modulated by the underlying structural connectivity. And some of the findings, early findings, suggest what I just mentioned earlier before, and that is that even though there isn't a strong causal relationship, the activity that is elicited during functional connectivity seems to be modulated somehow by the large-scale anatomical structure of the cerebral cortex. When you look at the results of some of these publications, what you notice is that it's not perfect. Some of the features that you observe in resting state functional connectivity can be reproduced by the model, but there are some salient problems, some of which could be related to the way in which the fMRI studies are designed, the issues with the limitations of the technique used on this particular publication for mapping connectivity. Specifically, there are some lateral medial connections in the parietal lobe that are known to exist that were not reproduced during this study on the structural part, and this have an influence on the observed functional connectivity of the study. So taking all this together, what we think is needed in order to be able to use the metaphor is to basically improve the accuracy of the diffusion model used for developing the structural connectivity, and also somehow trying to improve the quality and or specificity of the functional connectivity data. Now, on this latter aspect, there is recently a paper out of Yale University that appeared in Nature Neuroscience that basically demonstrate that even though previous reports seem to indicate that functional connectivity exhibits a high degree of variability between subjects and between sessions, if you take this data and you break it down and identify some very specific functional connectivity networks in the brain, you can actually begin to identify individuals, and not only can you do that, but you can also, in addition to that, predict the performance of the individuals on very specific cognitive tasks. And so this kind of underscores the fact that if you do the analysis right, there might actually be signaling there that can be used to try to develop what could potentially be a biomarker. Now, from the diffusion point of view, we know that the current state of the art with diffusion, prior to some of the work that I'm going to describe to you today, has some limitations. There are a number of techniques that have been, over the years, introduced to try to remove some of the problems. The first one, obviously, diffusion tensor imaging, which has been around for a very, very long time, is very limited. We've known that for a while. And in order to try to remove some of those problems, a technique called cue-ball imaging was developed in the late 90s, early 2000s. And that technique improves over it, but it has a poor mathematical representation of the diffusion model, and therefore still has some limitation. And I will illustrate what this looks like in a second to you. So the techniques that we're really going to focus on today is diffusion spectrum imaging, which was the first inception of a technique that has all the capabilities that you will need, but it has some limitations in terms of accuracy and speed. And then after that, in order to solve that problem, we developed this technique that we call radial diffusion spectrum imaging, which now is the basis of what we use here at NYU. And so just quickly to illustrate to you the problems with the previous technique, the structural connectivity has been used for surgical mapping extensively. And the images on the bottom side of the figure illustrate to you the problems that the techniques such as diffusion tensor imaging have had in the past. Namely, it cannot trace crossing fibers within a voxel, and as a result misses connections going, in this particular case, the projections from the corona radial to the motor cortex. So to solve this problem, essentially you need to look at the problem that you're trying to solve and what you do to basically properly map this out. And the way that we do this in MR, when we do diffusion, is that we collect a set of images in which we synthesize or sensitize the signal intensity at every voxel based on the direction of the preferential direction of motion that the water has at that particular voxel. And then what that means that if we look at an image, then for every given voxel within the image, we're going to have a series of data points. corresponding to the number of times that we collected that particular slice. And these data points are going to vary depending on the sensitivity of the motion at the direction that we were interrogating. Then we can take this data, put it in some genetic space that is related to the direction in which we synthesize the motion. And from there, we can then do a mathematical analysis from which we can develop what we call the orientation distribution function of ODF, which is that little glyph that you see there on the right. And what that little glyph represents is basically the preferential directions of motion for the water within that voxel. And in this particular case, that would represent a voxel in which we see essentially 3 crossing fibers. When you do this across the entire brain, then you get a map that looks like this. And from this map, it's fairly obvious that you can see some long-range orientation, preferential orientation in the water motion within the brain. But you also see some problems, like this one that you see in here. That little star that you see in there is basically not real. And I will explain to you how we can actually remove that later on. This is one of the problems that we actually had to overcome before we embarked on the project here. But then when you do that, and then you basically assuming that preferential directions of motions are continuous across space, then you can start doing tracings, and then you get a result that looks like this, that is essentially what we call a tractogram, or sometimes I'll call it a noodle map, for reasons that you will see later. Now, these maps have been used for quite a while. The technique was actually originally introduced in 2005, but at the time it required about 45 minutes for scanning, and so most people did not really use it. However, when you do use it, you find that it's actually much more accurate than anything that we had seen before. And this is an example of how it can actually very accurately map complex three-dimensional tracings. You have a triple crossing here in the central Semio Valley. It can also map complex turns, like in the Meyer's loop, which is basically 180-degree turn in the direction of the fires, as well as a crossing from right to left in the optic nerve. These are very challenging for other techniques, but the techniques can handle that quite well. And it also, you look at the terminations of the tract that you actually trace with this technique, you will find that these terminations follow the cortical anatomy of the brain as well, which is also a good indication that of its accuracy. So with this at hand, then you can start asking questions about long-range cables, quote unquote, in the brain. And for example, you look at the Arcuate fasciculus, then you can interrogate differences in individuals, and this is a comparison between a normal control and the brain, the Arcuate fasciculus of Temple Grandin, which is a very well-known autistic personality. And since in autism, part of the problem that you have is related to speech, when you look at the projections onto the speech cortex, what you actually notice is that this artificial sprouting that you see in this condition seem to basically produce a very inefficient innervation of the cortex, and therefore that could be, in a way, hypothesized to be related to the condition. We have also seen that this technique can be used for looking at structural disruptions in connectivity as a result of a concussion. And this is an example of a normal MRI scan for a professional football player. And when you do the diffusion spectrum imaging, you can very clearly see the fiber disruption in there. Developing measures with this technique for trying to diagnose conditions, that has also been explored. And this is an example of a paradigm used at the University of Pittsburgh, where I was before, in which essentially to try to detect abnormal connectivity, you compare the number of count of traces, the number of tracks that you have on the corona radiata going left to right across the two hemispheres. And on the normal control, these two maps should be nearly identical. And when you get somebody with a traumatic brain injury, you can actually see that the same map illustrates that there is a lack of connectivity or some disrupted fibers in one part of the corona radiata, and these seem to correspond very well with the motor deficit that this particular individual had. So this kind of like quick overview illustrates to you that the fact that this technique is actually good for finding variations in individuals. albeit these variations are actually quite severe. And so what we want to do is potentially look at more subtle variations, and therefore we need to look a little bit more closely at the accuracy of the technique. And so going back to this problem, we needed to basically resolve this problem. And as you're going to see in a second, this problem was related to some of the numerical artifacts that can be generated when you're using a technique that doesn't use the data that you acquire very efficiently. And so a very quick way to illustrate this is to use an exact two-dimensional model in which we simulate the acquisition of the data for two crossing fibers in 2 dimensions, indicated there by blue. And then here what you see is that if you try to use what's considered to be the state of the art right now, or what's mostly used by everybody, you can certainly see that you have two crossings going here in these 2 directions, but the severity of these crossings, or how pronounced they are, is really compromised by some of the limitations of the technique. If you then use diffusion spectrum imaging, you notice that you can actually see the directions, but you also have these artifacts in here, which are essentially the mathematical explanation of what you saw on that little star glyph that I showed you before. And then if we use the technique that we developed here, you notice that essentially not only can you see the fiber, but these artifacts are completely removed. And so after we developed this, we proceeded to actually evaluate this in not only in 2 dimensions, but also in 3 dimensions. And this is a 3-dimensional simulation as opposed to 2D. And here you can see, as before, that when the acquisition parameters are those that correspond to essentially an average scanning platform, you can actually have the kind of artifacts that I showed you in the previous image when you use the conventional DSI implementation. If you use our implementation, the artifacts disappear, and obviously, as you increase the aggressiveness of the acquisition, things get much more better for both of them. And so we went ahead and compared this in controls and basically perfected the way that the acquisition is done, and did some validation using bootstrapping to demonstrate that not only was the technique able to reproduce what we knew about the brain via DSI, but that actually the rendition that we get with it is actually more accurate. And this, in this particular case, corresponds to the areas where you see blue here. Blue corresponds, generally speaking, to lower error. And so you see that the Radial DSI technique in general is much more accurate. And so with that at hand, we developed a protocol, and for collecting the data that we're going to show to you, these are some of the relevant parameters for the diffusion acquisition. And then after collecting data on a number of volunteers and control, we decided to start taking a look at the data and then trying to understand how the higher dimensionality of this data could be used to our advantage. And so the first problem that we found is that the conventional assessment of diffusion measures using what's available in the neuroimaging community doesn't really utilize the multidimensional nature of the ODF. It's really designed for doing diffusion tensor imaging, which is a much more lower end technique than what we have right now. So what we did is that we took data like the one that we showed you before on a single individual for which we collected data multiple times, so we can do bootstrapping. And then we simulated 2 different groups of subjects where we expected to see differences. And so the idea was we can pick voxels from the brain where we know that the diffusion should be identical, and that therefore the variations in our estimate of connectivity at that particular voxel are going to be related to noise. And then within the same individual, because we have multiple instances of the data, we can then look at 2 different voxels that we know are very different, and then see how the detection power of the statistical analysis that we're using changes as we increase we vary the parameters of the acquisition. And so, case in point, if you look at a tractogram from DSI, so what we were interested in doing is confirming the null hypothesis. So in that case, we will basically have 2 voxels, 2 samples for the same voxel, or 2 distributions for the same voxels. In this particular case, will be at the genu of the corpus callosum. And then if we wanted to reject the null hypothesis, we will use two voxels that we know look different. For example, the one at the genius of the corpus callosum and one just anterior to the SMA. And so this is the data that, this is what the data look like. And what we basically found is that the statistical test that was probably better suited for looking at this was actually using a principal component analysis, because it emphasized the use of the higher dimensionality of the data and was better able to separate the conditions that I just described to you. With that in hand, we then decided to see what these findings correspond to in the subject pools that we had. And so to do that, we basically did a PCA analysis on a voxel-wise PCA analysis across the entire brain for the two groups. And one of the areas that appeared to show up was essentially the precuneus. This area corresponds to an area where people have reported previously that you actually do see changes. And this is just an example of what the data look like. And this is comparing TBI versus control. You can see for this particular collection of data, the significance is actually quite high. And just to demonstrate the necessity to do the calculation correctly, if we take this same data and instead of doing the radial DSI analysis, we use a QBI or a Q-ball imaging analysis. Essentially, this is what happened to your data. The significance essentially goes out the window. And then we were also interested in trying to find out what these changes meant anatomically. And so one of the questions that we asked is what happened in that part of the brain. And so we initially took seeds in the pecuneus, focusing on that particular area for the moment. And then what we have observed is that when you look at the mean length of the tracks crossing to that particular area between these two groups, they appear to be shorter. So once we had that at hand, we were interested in actually seeing, doing some more sanity checks in this particular data, seeing if this particular set of results actually stood when we look at it with a slightly different and perhaps more powerful tool. And to do that, what we decided to do is to borrow some techniques that have been used in the dynamic imaging and video compressing field. And that is the concept of low rank plus sparse decomposition. And the idea here is that we're going to try to be looking at features across the subject population that are common to them, and we're going to try to find a way that is very specific and very powerful for identifying essentially individual variability. And so if you look at this video, what you're going to see here is a set of video frames of people walking through a mall. And what this technique is very good at is identifying what parts of the visual field are basically stationary, i.e. they have temporal correlations, and what part are actually changing, i.e. are sparse or correspond to individual variability. And so when you look at the video, what you're going to see is that if you consider each frame as a matrix, essentially this technique is very useful for essentially looking at what changes and identifying what doesn't change. And so the metaphor here, or the paradigm that we're going to try to use, is that essentially if you think that two different scenes corresponding to two different pictures of two different malls correspond to two different groups, then what's common across groups should be the low rank part of the scene, and what's different across groups should be the individual variability that you have within the groups. And so, put it mathematically, we're assuming that our sample matrix now is going to be corresponding to a low rank component and a sparse component where most of the individual Sawyer variability is going to be captured. And this is going to be the same for the two groups, and therefore the difference between the two groups is going to be what's contained in the difference between the low rank components of this. And so to test whether this worked in this setting or not, we decided to do a set of simulations, again, to try to test whether mathematically this actually makes sense. And what we did there in that case is that we generated data for two ODF, orientation distribution functions, And then we took one of those and we can vary one of the diffusion parameters, which could be either the diffusivity or the angle. And this could be very basically according to whatever magnitude or change we wanted to. And here in this particular example, we have 10, 20, and 50% changes. And so what you notice here is that, so we can generate this data, and after we generate the data, we can add noise to it. We can do that about thousands of realizations of the noise, and effectively end up with a Monte Carlo simulation that allows us to statistically assess how this decomposition is helping. So for the 10% change in the diffusion parameters, what you see here is that the standard linear approach, i.e. PCA, which is a linear decomposition, basically cannot detect the change that we actually introduce. However, if we use the decomposition that I just showed you, we can actually detect it with a significance of 0.02. The minute that we take the changes in the parameters and we increase it to about 20%, then the significance actually jumps quite a bit, to 10 to the minus 10. And at a 50% change in these parameters, then the significance is extremely high. And this is essentially what you would need in order to be able to see it with the standard linear approach, or PCA. So, given that this appears to give us a little bit more power and the ability to reject outliers, we decided to then take the same data that I showed you a minute ago and then evaluate it in this setting. And so to do this, what we did is that we parcellated the brain into a number of structurally relevant ROIs, and we're using the AAL atlas for that, which has about 116 different anatomical regions in it. Then for each one of these subjects, then we calculate the connection indices, and these are basically the number of fibers between ROIs, the apparent fractional anisotropy between the two ROIs as we go to the fibers, and the quantitative anisotropy as we cross the fibers between the two ROIs. And we did that for every pair of two ROIs that can be computed. And from there, we develop what we call connection matrices. And then we can take these matrices, we can do the L plus S analysis of these matrices, and then we can threshold that to different significance levels to try to see what is different and or common across groups. And so here is a picture of the atlas that we use for this purpose. And this is what a typical connection matrix looks like. And in this particular case, the parameter, the diffusion parameter that we are showing to you is actually the fiber length. And so here, if you look at every dot in this matrix, it corresponds to the average length of the neuronal tract between the corresponding ROI indices that you have in here. So we go through the indices, we go to different places of the brain. The matrix is obviously symmetric, and then higher values, warmer values correspond to a larger number of fibers connecting those two ROIs. And obviously, if you go through the diagonal, then the value should be 0 because the connection there are no fibers between an ROI and itself. So now, once you have these fibers, these connection matrices, and you can do this, like I said, for different diffusion parameters. It could be the length, it could be the functional anisotropy, or it could be the quantitative anisotropy. And then if you think of a matrix, again, as a scene, then what we can have is that for every subject, we have a scene, or a picture of the scene that we were looking at before, and then we can actually collect this data for every subject and make it into a larger matrix, where now every column that you see here corresponds to the data observed for each one of the subjects. And then we can essentially do the same analysis, and we can try to see for different settings in the algorithm how much of the individual variability we're able to retain. And so one of the tuning parameters in this particular algorithm is basically the regularization parameter called lambda. And what you can see over here is that when the value for the parameter is low, then what happens is that essentially you are basically removing all of the variation, and essentially you are averaging out over the entire data set. So individuals are across this way. Every column is an individual. And then as you start relaxing the regularization parameter, then essentially what happens is that you are allowing more and more variability to come through into your low rank decomposition. What we have found empirically, although there is a mathematical way that you can actually set this, you can empirically figure out more or less what level of variance you are willing to accept. And in our case, between 100 and 200 seems to be the magic number. And so once you do this decomposition across the subject pool, which will be containing in this essentially long scene, then you can then look at the low rank component of the matrix, and you can actually do voxel-wise statistics on that matrix. And you can threshold that to a particular significance level and see what's left. And so this is exactly then what we proceeded to do with this particular subset of the data. And you look then again at the mean fiber length, that is the length of the fibers connecting two different ROIs. This is the map that you obtain. This map basically corresponds to those areas where the connectivity between those areas is different between the two subject pools that we're talking about in this particular example, which will be essentially TBI and control. And interestingly enough, it appears that the connections to and from the precuneus appear to basically be abnormal or different between these same subject populations. A couple of other things to note is that areas in the inferior temporal lobe and the medial temporal lobe appear to light up, although we don't feel very confident that this particular finding might be real. And the reason for that is that the diffusion images that we normally use have severe amount of distortion in this area. So on an individual and or group level, you're going to see a strong variability in the images in this area that we don't think has anything to do anatomically, and it has most to do with the ability of the magnet to produce undistorted images in this part of the brain, which is a known fact given the closeness of that particular location to the auditory canal. All right, so then in summary, what we've been able to see in this preliminary assessment of the data available through the CBC is that it is obviously clear that the use of Radial DSI increases the accuracy and the speed of the diffusion spectrum imaging methodology. The differences in tract connectivity in the precuneus are consistent with the etiology of TBI and PTSD, and they appear to basically overcome two different passes of statistical analysis that we have carried out on the group. The next question, and obviously the most interesting one, is how we can actually take this and move away from the subject, from the group analysis to a single subject analysis. And we feel that using a decomposition very similar to what was used in that paper from Nature Neuroscience that was published last year, we might be able to achieve just that. Although we need to take a closer look at the data to see what combination of connection indices may actually be the one that give you the specificity that we're looking for. With that, thank you very much, and I'll be happy to answer any questions you might have.
All right, thank you so much, Dr. Boada, for a very interesting talk. We'll allow a couple minutes for people to submit questions using the Q&A button. It doesn't look like there's anything in at this time, but please feel free to write something in. I would just like to remind everyone that this webinar will be archived and will be available on our YouTube channel and on the website, so please encourage your friends and colleagues to view it. And if you have any questions, please submit them now. So Dr. Boada, I was just wondering, do you think you can identify a specific pattern of decreased neural anatomical connectivity that would be specific to PTSD patients relative to other patients who may have similar decreased connectivity?
Well, I mean, that's our hope.
Hope point here.
Right, but I don't think that with the data that we have right now, we know what that particular combination is going to be. If you recall what was done in the Nature Neuroscience paper, essentially what they found is that when you look at the patterns of functional connectivity in the brain, you have a large collection of functional areas that light up in a very specific pattern for every individual. When you take that and you decompose it into individual networks, or basically on a smaller subsets, it is only one subset of those that appears to be strongly correlated with a particular individual. There are some theories as to whether or not this has to be with the anatomical variations of the individual, or whether this is in fact somehow related to the functional performance of that particular brain. And so there is still work going on in that area. What we feel is that to the extent that that trauma causes a rewiring or disruption of the wiring of the brain, this has to be a subset of the normal anatomical structural connectivity of an individual that could be correlated with some of the symptoms that a particular individual presents. And to get there, we're going to have to basically again increase the size of the data set and try to look deeper at the correlation between the different connections that you can establish and the symptoms, and then we'll see what we find.
All right, very interesting. Well, it appears we have no questions at this time. So Dr. Boada, I'd like to thank you on behalf of Dr. Magali-Haas and everyone at CVP for an interesting and engaging talk, and really appreciate your time for this. And also thank everyone for their participation. If any additional questions come in, please do not hesitate to contact us here at Cohen Veterans Bioscience. And thank you all for your time.
Thank you.
Thank you.
Bye.